Okay, hello everyone at NF Midwest and anyone else who might be dialing in. My name is Matthias Karajannis. I'm the chief of the Pediatric Neuro-Oncology Service at Memorial Sloan Kettering Cancer Center in New York. And I've been involved in NF2 research, and I've been also taking care of NF2 patients for close to 2 decades now. And it's been an amazing journey and a very rewarding experience. So today I was asked to present a talk on laboratory models of NF2 related tumors and how they inform clinical trials. And I am happy to do so, and I'd be happy to also answer any questions that you might have at the end of this presentation. I try to keep it basic in the sense that it's geared to be understandable by non-specialists and non-experts, meaning patients, as well as individuals affected with NF2. So thank you for your attention. So here are some disclosures for activity on as a consultant and on medical advisory boards for pharmaceutical companies. None of this is directly related to the information I will be presenting. So as many of you know, NF2 or NF2 related schwannomatosis, as it is now called, is an inheritable genetic disorder and tumor predisposition syndrome. And as you can see here in this cartoon, we have 2 copies of the NF2 genes, also called Merlin, one on each chromosome. So we all normally harbor 2 copies of this gene in our normal cells. And individuals with NF2 will have lost or inactivated one of those gene copies, either through a point mutation or through a larger deletion of that gene, but will retain a second normal copy that's shown on the top. And then when the second normal copy is lost, and that usually happens either through a second mutation or through what we call a loss of heterozygosity, meaning there's a larger deletion of parts or all of the chromosome, a cell will wind up with no functional copy of the NF2 gene. And if that happens at the right time in the right cell, a tumor will develop. And we call that loss of function or a tumor suppressor, meaning that when that tumor suppressor is lost, which normally suppresses growth and suppresses tumor formation, a tumor will occur. So over many years, really over the last about 2 decades, a lot of researchers have worked on deciphering what happens when NF2 or Merlin is active in cells and how it works as a tumor suppressor. And fortunately or unfortunately, Merlin has a lot of different functions in the cell, meaning it interacts with a lot of different proteins, and this is summarized here in this cartoon. So what you see in this cartoon, all of these circles or ovals represent proteins. Most of them actually represent kinases, meaning enzymes. And the Merlin that is normally present in cells will interact, typically inhibit the function of many of these proteins and kinases. And conversely, when Merlin is lost, many of these kinases will be overactive, and that is how we believe Merlin acts as a tumor suppressor. And this has opened opportunities for therapeutic interventions because many of these proteins and kinases that Merlin is interacting with and that are hyperactivated when Merlin is lost are now targetable by specific drugs that inhibit these kinases. Now, how do we, how has, this type of signaling been worked out, and how can therapies such as drugs, including kinase inhibitors, be studied in the laboratory? Well, first, one needs to have a model of relevant cells. And so for NF2, the relevant models would be models of schwannoma or meningioma, and to a lesser extent, ependymoma. And one way this can be done is to use fresh human cells, and you can culture them in the laboratory using specialized techniques. This would be called in vitro work, meaning that you take cells out of a individual or out of an animal, and you culture them in, so to speak, in the petri dish. That is called in vitro preclinical testing. And so you can see examples here on the left side. This is a microscopy image of primary human vestibular schwannoma cultures. So these were vestibular schwannoma cells that were obtained during a surgery. And then these cells were cultured in a special medium. And they will continue to grow under those types of conditions. But there is a major limitation in that the growth is relatively slow. And that makes sense because these tumors in humans also require years, sometimes decades, to grow. And the other problem, or the other limitation rather, is that they will not keep growing forever in culture. After a few cell divisions, the cells will exhaust themselves and eventually die because they're not growing in their natural environment. And they don't receive sufficient signals to stay alive for a long period of time, which really limits the ability to study these cells. On the right side, you can see an example of fresh meningioma cells that were harvested from a tumor specimen after surgery. And it works very similarly. So you can culture these cells in the laboratory under specific conditions. The cells will continue to grow for a while. They will undergo a few cell divisions, will continue to grow for a limited time. And one can do experiments in these cells. One can look at proteins, one can look at drug exposure, and so on. So these cells are, very useful. But again, the limitation is that the time over which these experiments can be done is very limited. These cells will only grow for a limited amount of time. And of course, there's also limitations because these cells have now been removed from their natural environment or microenvironment. So all of the other things that inform the cells, that help the cells grow, that are relevant for the biology of these tumors, for example, that includes immune cells, that includes support cells, that includes vascular or blood vessel cells, and neurons, nerve cells, and so on. So there are significant limitations to these in vitro fresh human cell models, but there are also some advantages in that they are relatively easy to obtain, and they have not been tampered with, so to speak. So these are, still genuine human cells that have not been altered, and I'll get to that in a moment. Now, here's an example from a study that we published 14 years ago in a collaboration with Dr. Hahnemann's lab in the UK. And here's an example of using fresh human cells and learning about the biology and learning about possible treatments. And in this case, what you can see here on the right, we studied a drug called lapatinib. And we showed in this study using fresh human cells that when you expose them to lapatinib, that you reduce the growth of these cells, and that has led to a subsequent clinical trial. So this is one example where we have learned a lot of important information from using fresh human cells of schwannoma cells in the laboratory and translating that into a clinical trial. Lapatinib is a kinase inhibitor, and so this is an example of targeting a overactive kinase in NF2 deficient cells. Now, I told you that the problem with using fresh tumor cells is that these cells exhaust themselves after a relatively short period of time, which limits the ability to study those cells. And so one way to get around this is to use immortalized schwannoma or meningioma cells. So these are cells that are sometimes commercially available. And in this case, you can see in the right lower corner, immortalization method, SV40 large T antigen. So these are cells, in this case, schwannoma cells, that were manipulated by using a viral vector and inserting foreign DNA, basically, into those cells, and thereby immortalizing them, meaning they will continue to grow essentially forever if they are cultured properly, and they can go unlimited amounts of cell divisions. And that is, usually something that characterizes cancer rather than low-grade tumors, such as schwannomas or meningiomas. So basically, you're turning a low-grade tumor cell, like we see in NF2, into a cell that is more similar to a cancer cell. And the advantage is that, as these cells continue to grow, they're easier to study. They tend to also grow faster, so you can get results quicker. But of course, the drawbacks are that now you have cells that are somewhat removed biologically from the original type of cell. They have a different growth rate. They have some different functional properties and biological properties. So there are, of course, some drawbacks, and that's the case with every type of model that we use preclinically in the laboratory, that depending on the model, there's always advantages and disadvantages, and none of them replicate exactly what happens in the human body. So now I'm going to switch to in vivo testing. So we talked about in vitro testing, which is basically growing cells in a dish. And that is one way to study cancer cells or tumor cells, and it's very useful. But as I told you, you are missing, by using those in vitro models, a lot of the parts that normally form a tumor, which is not just composed of the tumor cells themselves, but also all of the support cells, immune cells, vascular cells, nerve cells, and so on. So in order to study tumors, not just tumor cells in mice or other animals, there are different ways to go about this in general. So one is by removing a piece of tumor, either from a human tumor or from a tumor that grew in a mouse. So you can see that on top under A. And then you can dissociate those cells, or sort of, chop them off, in the dish. You can then transform them potentially, because the problem is that if you use a low-grade or quote benign tumor cell, for example, from an NF2 schwannoma or meningioma, and you transplant this into a mouse, the cells won't grow because they're not malignant enough. And I told you that, after cell divisions, they will die off because they're not in their optimal environment. So one way to go about this is you can use immortalized cells, like in the example I just showed you. So you can use cells that have been perturbed by genetic modifications, thereby making them more aggressive, making them immortal. And when you transplant those into mice, they will grow, and they can be used. So they can be used to study, for example, schwannoma or meningioma tumors in the mice. It's important to note that these mice have to be immunocompromised, so they are genetically modified to have a abnormal or very weak immune system, because mice with a normal immune system typically would reject cells that are transplanted that are foreign. So typically these experiments are done in mice that are immunosuppressed, which has the disadvantage that the immune system is largely absent in these mice. And therefore, if you want to study the effect of the immune system in tumors or on tumors, you can't really do this. And again, is a limitation of this particular model, because these cells have to grow in severely immunocompromised mice. Below in B, you can see a different, slightly different approach where you directly put cells from either a human tumor or a mouse tumor into an animal, and that would be called a patient-derived xenograft. So you put a piece of tissue of tumor directly from one individual or animal to the other. If it's called, if the tumor is transplanted from the same type of species, so for example, from mouse to mouse, we call it an allograft. If it's transplanted from a different species to another, so from human to mouse, we call it a xenograft. So in this case, we call these tumor models patient-derived xenografts if the tumor comes from a patient and is put into a mouse. And again, this will only work with mice that are genetically engineered to be severely immunosuppressed, because otherwise they will reject the tumors, number one. And then again, number two, this typically does not work with slow-growing or benign tumors as you have in NF2. It typically only works when you have cancerous or malignant types of tumors that are growing fast enough and that are already immortalized by default. Now, another even more sophisticated model for preclinical testing in vivo or in the animal is using genetically engineered mouse models, or GEM. So the way this is done is that you have a mouse and you specifically genetically engineer the mouse that you can turn on an oncogene or you can turn off a tumor suppressor. So basically you have a very targeted way of turning on or turning off a particular gene in a particular cell type. And when you do that, the mice will develop a specific type of tumor that is driven by a specific mutation, either loss of a tumor suppressor or by gain of an oncogene, so by an oncogenic mutations. And sometimes these models require or work with multiple types of mutations. Sometimes one type of mutation is enough in the right context to form a tumor, but this is a very specific genetically defined way to generate specific types of tumors in mice. And there is an example for NF2 where the mice are genetically engineered to express NF2 in all of the cells, except for certain types of cells, there is an additional gene inserted. So when these mice are fed a specific antibiotic, there will be a genetic rearrangement happening in a particular type of cell. In this example, these are cells that express a protein called periostin. And what happens is that this is a targeted way to induce a loss of NF2, just like it happens in human tumors, in specific types of cells in the mouse. And in this case here, what is shown is that when you do that, the mice develop schwannomas, either in the vestibular ganglia, so this is in the hearing apparatus, similar to vestibular schwannomas, and or in the dorsal root ganglia. So these are parts of the nerve that come off the spinal cord, also similar to what we can see in NF2 patients. So this is a genetically engineered mouse model that very specifically will inactivate genes in certain cell types and de novo, or as new, will generate tumors in the mouse that resemble the human tumors. They have similar biology because they also have lost NF2. And the advantage of this type of model is that it does not require an immune suppressed mouse, because the tumor cells have otherwise the same genetic background as the mouse itself, so it won't reject those types of tumors. And in many ways, these types of tumors more accurately reflect what is happening in the human equivalent disease. And you can also use this model, like the other models, to study the biology of these tumors. And you can also study what happens when you expose these tumors to certain drugs or other interventions. And how do you look at what happens when you induce these tumors, or when you treat these animals with certain drugs? Well, it's a little bit more complicated than looking at the cells in the dish, where you can just basically count them or study them relatively easily. So in these animal models, left upper corner, you can see an example of these dorsal root ganglia in the model that I just showed you. So you can remove them after the tumors have grown for a while, and then you can dissect them, you can study them, and you can compare the growth of treated root ganglia versus non-treated root ganglia, etc. So that's how you can study the tumors from this type of model. And then there's another very elegant way how you can monitor the size of tumors in animals without having to actually get the tumor, or resect the tumor, or remove the tumor, or measure the tumor directly. And the way this works is called chemiluminescence. So you can introduce an additional gene into the tumor cells. That can be done by genetic engineering of tumor cells themselves, or it can be also done by gem models. And what that does is that there is these specially engineered tumor cells will emit a light that can be captured with a special camera. And by doing this, one can measure the size of the tumor as it grows in the mice. So there, in this example here, you can see that where these circles in different colors measure intensity of the light and the size of the tumor. And in order to do this, you have to anesthetize the mice temporarily so that they don't move, and because taking these pictures takes a while. And here's an example from a study that one of my basic science collaborators, Joe Kissel, has published using a drug called crizotinib. It's a kinase inhibitor against a kinase called focal adhesion kinase that is overactive in tumors that have lost NF2. And he showed very nice biology findings using this drug, but also tested in animals, as you can see here. You can see on the right side, the animals that were treated with crizotinib had less tumor growth than the mice that were treated with what we call vehicle or control. Is basically they were administered a carrier without the active drug. And this has led to a clinical trial that I'm chairing in the NF Clinical Trials Consortium that is currently ongoing. And the drug is similar to another drug called brigatinib, which is also a FAK inhibitor that you might have heard that has been used also in clinical trials that were recently published, specifically in the Intuit clinical trial. So here's an overview, and this comes of clinical trials that have been performed using drugs. Most of these were kinase inhibitors. And this original list is from a review article that we, meaning NF2 specialists in the field, wrote back in 2009. And many of these proposed drugs came out of preclinical data and were felt to be good candidates for translation into clinical trials. And what you can see marked here in green are drugs that were actually then subsequently tested in preclinical trials. You can see Avastin or bevacizumab here at the bottom left. And all of the other drugs that were used here have really been kinase inhibitors. Avastin is a monoclonal antibody. And what you can see in green are the results of these studies with PR, meaning that partial responses were seen, meaning that tumors in NF2 patients when treated with these drugs did not disappear, but they shrank. We call that a partial response or partial shrinkage. Or trials with drugs that showed stable or suggested stable disease, meaning they seem to slow down growth of tumors. And so here are examples. I showed you lapatinib earlier. This is a drug where we saw partial responses in a subset of patients with vestibular schwannomas. Alatinib is a similar drug that was studied with similar results. Everolimus or RAD-001 is a drug that was studied that primarily seemed to result in stable disease. You know about bevacizumab, very likely. And there are some other studies, some of them which have not been published yet. And there are recent and ongoing studies with other kinase inhibitors. I mentioned brigatinib as well as crizotinib. So these are FAK inhibitors. And a new drug or a new agent, neratinib, is also now in clinical trials in the Intuit clinical trial platform. So when we think about designing these clinical trials and taking into account what we know about the biology of these tumors that I told you about, is that we have to consider how efficacious is the drug versus how feasible is the drug. And ideally, we would like to have a drug that is highly efficacious, meaning that works well in terms of shrinking tumors or at least stopping their growth. But it also has to be feasible in the sense that the therapy can't be too toxic or can't have too many side effects, because that would, of course, limit the feasibility for patients to use that drug, to tolerate that drug, and to have an acceptable quality of life, because all of these drugs have undesired toxicities or side effects. And that goes really for any drug, not just for cancer drugs. And so when one thinks about this conceptually in terms of, what would be an ideal drug to treat patients with NF2 related tumors in particular, but really for any tumor in general, would be that ideally it would be a medication that one can take by mouth and doesn't have to be administered IV. It would ideally have no toxicity or low toxicity. Ideally, it would treat all tumor types that we see in NF2, schwannomas, meningiomas, and ependymomas. Ideally, one would see high response rates, meaning that all tumors would shrink or most tumors would shrink, and hopefully by a lot. That would be a major volume reduction. We would also like to see a functional response, as we've seen with drugs such as bevacizumab, where functional improvement can be accomplished. In that case, a hearing improvement can be seen. And then ideally, the treatment would be durable, meaning that when the treatment is finished, the effects would still continue so that you wouldn't have to be on that treatment for the rest of your life. And as you probably know, unfortunately, we don't have this type of perfect treatment or drug available for NF2. So everything that we have is a compromise in some or all of these areas. So we have drugs that are given oral, but they have moderate toxicity that treat some of the tumor types, where we have some response rates, where we have some volume reduction, but tumors don't disappear. We have some functional response, but the response is really not durable, meaning the benefit is only seen as long as you are on the drug, and that goes for most of the drugs that have been studied in NF2. And why do we, why do we see that, and why do we have to deal with this compromise? Well, that has to do with what's called the therapeutic window in drug therapy. And that goes back at efficacy versus toxicity. So what you can see here in this graph, that when you move from the left to the right, as you increase the amount of the drug that is administered, so you increase the dose, of course, it gets more and more effective because the percent of patients responding are shown here on the y-axis. So as you increase the dose of the drug, you increase the efficacy, more and more patients or tumors will respond. But of course, the problem is that as you do that, you also increase the toxicity. And for every drug, the therapeutic index is, or therapeutic window is, how far is this efficacy curve away from the toxicity curve? And unfortunately, for many cancer drugs or anti-cancer drugs, such as chemotherapy, the toxicity and the efficacy curve are very close together. So there's a narrow therapeutic index, meaning that as you go up in the concentration and you get better tumor responses, you also very quickly get to a limit in terms of what a patient can reasonably tolerate. And so ideally, drugs that we would want to use in NF2 to treat NF2-related tumors would have a wide therapeutic window. But unfortunately, most of the drugs that or pretty much all of the drugs that we have been using in NF2 have a relatively narrow therapeutic index, meaning you can't give as much drug as you ideally would like to give to get the best response, because that's beyond what a patient could reasonably tolerate. And in terms of targeting cancers that are driven by mutations, there is a big difference between targeting tumors that are driven by a mutation in an oncogene, and in this case, an oncogene called BRAF. It's another kinase. So there's a particular mutation in this type of tumor in this kinase that causes increased cancer cell growth. You can see that here on the left. And this is fundamentally different than trying to target kinases in tumors that have loss of a tumor suppressor like NF2. because in this example here, there is a specific drug that has a very high affinity or binding to that mutant protein. It's about 1,000-fold higher than to the normal kinase. So it binds very selectively to the mutant kinase, and because of that, you get a very wide therapeutic, or relatively wider therapeutic window. And since these cells, in this case, low-grade glioma cells, are completely dependent and addicted to this mutant kinase, when you inhibit it, you get excellent responses. In many patients, tumors shrink by more than 50%, and in some patients, tumors even completely disappear, as you can see here in this so-called waterfall plot, where the amount of tumor shrinkage in percentage for each particular patient in this clinical trial is shown here. It is more difficult for when you try to target kinases, as I showed you, kinase proteins in patients, tumors who have lost NF2, because we're not targeting a specific mutation directly, but rather the effect, meaning that when NF2 cells have lost the NF2 tumor suppressors, they will overactivate kinases, and we can treat those kinases, and there will be a therapeutic effect, but it won't be as is predicted to be not as selective as in the prior example that I just showed you, where you have a drug that specifically goes after an overactive kinase with a very high affinity and leaves the normal protein largely intact. And so here on the left, you can see a waterfall plot from the response rate in terms of tumor shrinkage in tumors in patients that we treated with lapatinib. We published this back in 2012. These were vestibular schwannomas, and you can see, the majority of tumors had at least a little bit of shrinkage, and few tumors responded well, meaning shrinkage over 15 or over 20%. And there were some tumors in some patients that also did not respond. Of course, when we treat patients with these type of kinase inhibitors that I mentioned before, there's a limit in terms of the dose that you can reasonably give to patients without interfering too much with quality of life. And on the right side, you can see the recently published study from the Intuit trial, brigatinib. Target tumors are shown here, also in this waterfall plot. You can really see a very similar picture. These are, the way that tumors are plotted is just a mirror image, but you can see that, the bottom line is actually quite similar in that the majority of tumors will have, at least some shrinkage, but major shrinkage is difficult to achieve and happens only in a small subset of tumors. And, this experience using kinase inhibitors to treat patients with NF2-related tumors is really, shouldn't be too surprising, really, because it is really following the biology that I try to illustrate, in that treating kinases in NF2 mutant tumors, will have a limited therapeutic index, as we can predict from the biology and from preclinical trials. Now, I told you a lot about inhibiting overactive kinases in cells that have lost NF2, but there are other ways to treat NF2 deficient tumor cells. And one of them that I think is very exciting is treating the primary signaling pathway by which cells that are deficient in Merlin increase their proliferation and decrease their cell death. And this is shown here in this cartoon, where normal cells that have an intact Merlin or NF2 tumor suppressor the Merlin will act as a tumor suppressor, and suppress a transcription factor called TEAD, transcription enhanced associate domain. So this is a protein that we know is overactive in NF2 mutant cells because this pathway is overactive in NF2 mutant cells. And what happens is that this TEAD transcription factor, as a result, is overactive. A transcription factor is a protein that will bind to several areas, usually multiple areas in the genome, and will activate multiple genes as a result. And in the case of TEADs, will result in increased cell proliferation and decreased cell death. And we have data that's been generated for many years that this is a very important pathway that allows for NF2 mutant cells to grow uncontrolled. And it's a fundamentally different pathway than these kinase-activated pathways that I showed you previously. And these, as opposed to kinases, which are enzymes, these transcription factors generally are very difficult to target or inhibit with drugs. And that has to do with their structure. Kinases or enzymes are relatively easily targeted with specially designed small molecules or drugs. But for these transcription factors, which are large proteins that are not kinases, not enzymes, this is much more difficult to do. And many of these transcription factor proteins have long been considered, unquote, undruggable, meaning that it's very difficult or impossible to find a drug that will target those. But over the last few years, for some of these transcription factors, new drugs have been successfully developed. And this is very exciting in terms of inhibiting the TEAD transcription factors in NF2 mutant cells. And this was recently shown successfully in preclinical studies with one of the models that I recently showed you by a group in the UK, where not only decreased proliferation, but also increased tumor cell death was seen in mice with genetically engineered NF2 tumor-bearing mice that were treated with this experimental TEAD inhibitor. This is a drug, in that case, that has been developed by a company called Vivace, and they have run initial clinical trials for NF2 deficient tumors. And turns out that one of the cancer in adults that is the most, that has the highest prevalence of NF2 loss or NF2 deficiency is actually malignant mesothelioma. So they conducted a clinical trial for patients with cancers or tumors who had lost NF2. So you had to show it by genetic testing that the tumor had lost NF2. And they included those types of patients, but also other solid tumor patients. And they showed, as you can see in this plot, this drug seems to be quite promising for treating NF2 deficient solid tumors, including mesotheliomas. Because of the preclinical data I showed you on the last slide, and because of these encouraging finding that this drug truly seems to have anti-tumor activity, including or perhaps specifically against NF2 altered tumors, we are excited to translate this into a clinical trial with this TEAD inhibitor in the NF Clinical Trials Consortium to see if we can also see responses in patients with NF2 when treated with this drug, and to what extent we see responses. And if we can conduct this clinical trial successfully, we will see whether the types of responses that we'll see will reach or hopefully exceed the types of responses that we see with kinase inhibitors in NF2 patients. So in terms of going to conclusions, NF2-related tumors are considered a single driver, meaning they're only driven by one single type of mutation in one gene, but they're not single pathway, meaning that the loss of the tumor suppressor NF2 will activate many biological signaling pathways in the cell, and that makes it difficult to treat with drugs. Targeting these kinase enzymes is feasible, as I've shown you, including in clinical trials, but there's a limited therapeutic index, meaning many tumors will not shrink, and many tumors will only shrink partially. Differential responses in vestibular schwannomas versus meningiomas and ependymomas can be seen with these drugs. They don't work all the same in the same tumor type. And that probably has to do with heterogeneity, meaning that not all tumors are the same. There's probably problems with drug penetration into the tumor. Perhaps it depends on the tumor size and how well it is perfused with blood. Drug metabolism, different patients or different individuals will metabolize drugs differently. That may also be a factor. And then off-target effects, meaning that all of these drugs, including the kinase inhibitors, will not just inhibit one or two kinases that you're trying to target, but typically others as well, causing toxicity. We do see acquired treatment resistance despite genetic stability, which means that for patients that are treated with either kinase inhibitors or drugs like bevacizumab, what we tend to see is that after a while, it can be months or years, tumors will grow despite the treatment. And this is not because these tumors acquire new mutations, like cancers do often very rapidly. NF2-related tumors seem to be genetically stable, but they find other ways how to probably upregulate or deregulate expression of proteins in the cell, and therefore becoming resistant to treatment over time. And last but not least, I think we can be excited that novel, promising classes of drugs, such as the TEAD inhibitors that I've shown you, are recently being developed that will allow us to study targeting pathways and biology in NF2 mutant tumors that previously were not accessible, and hopefully will translate into clinical success. So with that, I will conclude. I'd like to acknowledge all of my active collaborators in the NF Clinical Trials Consortium, as well as Moffitt, Joe Kissel, and UC Florida, Christina Fernandez-Valle. I would like to thank the agencies that have supported my research, as well as companies supported clinical trials that I've been leading, and then as well as the fantastic team I have both in the pediatric department at Memorial Sloan Kettering, taking care of NF patients, NF1, NF2, as well as schwannomatosis, as well as on the adult side in adult neurology, adult neuro-oncology, and associated subspecialties. And last but not least, I would like to thank the patients that have participated in clinical trials that we have been conducting, and their families. Thank you very much.